Good evening, students. In the previous class, we have learned replication of DNA by semi-conservative method, experimental proof, which was given by Messelson and Stahl. So we learned Messelson and Stahl experiment to show DNA replicate by semi-conservative method. Further evidence was given by Taylor and his colleagues. Taylor and his colleagues in the year 1958. Okay, they gave evidence to support DNA replicates by semi-conservative method. DNA replicate by semi-conservative method. So Taylor and colleagues, they confirmed semi-conservative replication of DNA using appreciated thymidine. H3 tritium, radioactive hydrogen. Appreciated thymidine to detect the distribution of so they used appreciated thymidine to detect the distribution of newly synthesized DNA in the chromosome replication in the root cells of the plant, that is Vicia fava. It is also known as broad bean. So this was the quantitative experiment carried out by Taylor and his colleagues in the year 1958 to confirm DNA replicate by semi-conservative method. So they researched on the root cells of the Vicia fava, the chromosome replication, where how the newly synthesized DNA strands will be distributed. So that is what they detected by appreciated thymidine. So this was the confirmation, further confirmation given by Taylor and colleagues to support DNA replicate by semi-conservative method. Remember, for 2 mark, this can be very important for theory point of view as well as for need point of view. They can ask the plant name is Vicia fava. They use appreciated thymidine, radioactive thymidine. It is radioactive thymidine. Okay, to find out the chromosome replication. So how the radioactive level chromosomes will be distributed when the chromosome replication occurs. The daughter DNA, what will be the nature, labeled radioactive level or unlabeled. So that is what they detected and they confirmed DNA replicate by semi-conservative method. So this is all about the experimental proof or evidence. First given by Messelson Stahl, that is quantitative experiment. Then the qualitative experiment which was conducted by J. Herbert Taylor and his colleagues. Now the next concept now, requirements from DNA replication. You have to remember various requirements are there in DNA replication. So here various enzymes are required. See, the mechanism of replication has been investigated in E. coli, Escherichia coli, a prokaryotic cell, where this mechanism has been investigated. Later on, in the eukaryotic cell, higher mechanism like plants and in human cell also, it was investigated. So the enzymes which are involved in DNA replication, it is a multi-enzyme complex. Multi-enzyme complex can be involved in replication of DNA. So this we usually refer to as replication apparatus. Replication apparatus. So various important enzymes are there. The most important type of enzyme DNA polymerase enzyme. DNA polymerase enzyme. So in case of prokaryotes, three types of DNA polymerase enzymes are involved. These are mainly DNA polymerase 1, DNA polymerase 2, and the most important one, that is DNA polymerase 3. This is the most important one. These are the three types of DNA polymerase enzymes involved in prokaryotes like bacterial DNA replication or even in viral DNA replication also. So DNA polymerase 1. What is the role of DNA polymerase 1? DNA polymerase 1 is known as Wernberg enzyme. It is called as Wernberg enzyme. Okay, so this DNA polymerase 1 for the first time it was discovered by Arthur Wernberg. Discovered the enzymatic activity of DNA polymerase in the what we call replication of DNA. So it is referred as Wernberg enzyme. Remember, these two enzymes were discovered by Arthur Wernberg's son. Arthur Wernberg's son. Okay, now DNA polymerase 1. DNA polymerase 1, what is the role? It removes RNA primer. It removes RNA primer. We will come to know what is the meaning of RNA primer, all that. It removes RNA primer and fills the gap in between DNA strands. It removes RNA primer and fills the gaps in between the DNA strands, small strands which are there. That gap which is there, that gap can be filled. One row. It has even exonuclease activity. Exonuclease activity. This Wernberg enzyme, DNA polymerase 1, has exonuclease activity. Exonuclease, the nuclease enzyme which remove the nucleotide from the ends. So it has nuclease activity, exonuclease activity in this direction. That is 5' 3' direction, and even it can have exonuclease activity in 3', 5' polar direction also. Then it is involved in DNA repair. It is involved in DNA repair, particularly what we say proofreading. proofreading of newly synthesized DNA. In that, it is involved DNA repair, proofreading of DNA. In that, it is involved mainly in DNA repair. We'll come to know when we learn the mechanism. DNA polymerase 2. DNA polymerase 2. It is a special DNA repair enzyme. DNA repair enzyme. So it is also involved in DNA repair. It is also involved in proofreading of DNA work. Okay, the final stage. If any mistakes are there in the replication, that can be rectified by the activity of the polymerase 1 and 2. So DNA repair, what we said. The most important enzyme that is the DNA polymerase 3. DNA polymerase 3. So DNA polymerase 3, it catalyzes the replication of DNA in 5' 3' direction. So it catalyzes the replication of DNA in 5' 3' direction. So if this is a template strain, master strain, parental strain, if this is a template strain or parental strain, on this new strain will be produced in this direction. On this template strain, the new strain will be produced. This is the new strain. This new strain is produced in 5' 3' direction by the activity of DNA polymerase 3. DNA polymerase 3. Okay, catalyze. It is a very important enzyme. It catalyzes the replication of DNA in 5' 3' direction. So, in case of E. coli, the role of this DNA polymerase is very, very important. Okay, so DNA polymerase 2 and polymerase 3, Roger Kornberg, son of Arthur Kornberg. Roger Kornberg, son of Arthur Kornberg, who discovered the role of the DNA polymerase 2 and polymerase 3. This polymerase 3 is very important. It synthesizes the new strand of DNA in 5' 3' direction, and the template strand should be 3' 5'. So here, the role of these enzymes, particularly this polymerase 3 enzyme, is very important. These enzymes now, they catalyze the polymerization of nucleotides. Polymerization of nucleotides. Which nucleotides mainly? Deoxyribonucleotides. They catalyze the polymerization of deoxyribonucleotide. Several nucleotides are joined by phosphodiester bond to form a polynucleotide chain. So new strand is synthesized by the addition of individual nucleotide by the catalytic activity of DNA polymerase 3. Okay, so these are having what we call high speed of joining the nucleotides to synthesize the polynucleotide chain. When you take this E. coli, it has the base pairs 4.6 into 10 to the power 6 base pairs. Large number of the base pairs have to be polymerized to synthesize a new strand of DNA. So they have high speed and they have high degree of accuracy. High degree of accuracy. Usually, without doing mistake, they can add the deoxyribonucleotides. They can add deoxyribonucleotides without doing the mistakes, and they synthesize the polynucleotide strand. So they have high speed of addition of the deoxyribonucleotides and high accuracy they have. So in case of E. coli, to add these many base pairs, so it takes around 2000 base pairs per second. This is the speed of addition of deoxyribonucleotides by the DNA polymerase 3. So these many base pairs are there. These many base pairs are there, and the replication occur bidirectionally. Okay, so the time taken for the replication, okay, in case of E. coli, the time taken for the addition of these many number of base pairs to complete the replication, it is around 18 minutes. In 18 minutes, they may add up these many number of the nucleotides, and it will complete the replication process. So the speed of DNA polymerase 3 to add the nucleotides to synthesize the polynucleotide chain, that is 2000 base pairs per second. The number of the base pairs in prokaryote like E. coli is what we call lesser. Whereas in case of human beings, okay, the diploid number of the base pairs, you have 6.6 into 10 to the power 9 base pairs. The number of base pairs more in the case of human being. The diploid genome, haploid genome, you know, 3.3 to 10 to the power of 9 base pairs. This is haploid genome, this is the diploid genome. So in case of eukaryotes, the speed of DNA polymerase enzyme, there are different DNA polymerase enzymes are there. There are 5 types of DNA polymerase enzymes are there in case of eukaryote. The speed of addition of the deoxyribonucleotides by the activity of DNA polymerase. DNA polymerase there mainly what we call delta. DNA polymerase delta. In the case of eukaryotes, so there the speed of addition of the deoxyribonucleotides, it is 100 base pairs per second. It is comparatively slower because the eukaryotic DNA replication is much complex than the prokaryotic DNA replication that we remember here. Comparatively, the speed is more 2000, around 2000 base pairs per second, and the time taken for the replication to complete, it is around 18 minutes for addition of these many number of the base pairs in case of E. coli. So these are the 3 types of DNA polymerase enzymes in case of prokaryote. So DNA polymerase, very important enzyme which catalyzes the addition of the nucleotide. Nucleotides are simple monomeric units. These monomeric units are joined to synthesize a long polymer. So that is what catalyzes the polymerization of the nucleotides. Okay, so particularly in this direction, 5' 3' direction. On the template strand, that is 3', polarity of the template parental strand 3', 5', and new strand will be synthesized in 5', 3' direction. Remember this. In the case of eukaryotes, 5 types of DNA polymerase are required. DNA polymerase alpha, DNA polymerase alpha, DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, and DNA polymerase what you call epsilon. DNA polymerase epsilon. DNA polymerase alpha, it is carrying out initiation of replication. It is involved in initiation of replication. Beta, DNA repair. DNA repair. Then gamma, mitochondrial DNA. Mitochondria has a small double-stranded circular DNA. It is involved in that DNA replication. So mitochondrial DNA repair. Then DNA polymerase delta. It is involved in replication of DNA. Replication of DNA in 5' 3' direction. In prokaryotes, DNA polymerase 3 is involved. DNA polymerase 3 catalyzes the synthesis of new strand. Replication of DNA means synthesis of new DNA strand in this 5' 3' direction. So, in case of eukaryotes, you have DNA polymerase delta. Remember, DNA polymerase delta, which is equivalent to DNA polymerase 3 of prokaryote. Remember, DNA polymerase 3, what role it play? Same role DNA polymerase delta will play in case of eukaryote. DNA polymerase 1. DNA polymerase 1, which is involved in repair of DNA, here beta. DNA polymerase 1, it is involved in DNA repair in prokaryote, it is beta. DNA polymerase beta. So very important one, that is DNA polymerase delta. Remember, DNA polymerase alpha is involved in initiation, starting of replication. Okay, so in case of prokaryote, DNA polymerase 3. DNA polymerase 3 is itself involved in initiation. Okay, so this is involved in what I call synthesis of new DNA strand delta or elongation of the new DNA strand. Okay, so in case of prokaryote, initiation and elongation both are carried out by DNA polymerase 3. Initiation and elongation both are carried out by DNA polymerase 3 in prokaryote. In eukaryote, initiation of replication is carried out by DNA polymerase alpha, and delta is usually involved in what we call elongation. Then DNA polymerase epsilon. DNA polymerase epsilon, it is involved in polymerase activity of Halos cells. Polymerase activity of Halos cells. Halos. A American Negro woman. American black woman or Negro woman. Her name Henrietta Lacks. So this woman who was suffering from cervix cancer. Okay, cervix cancer. So the cancerous cells were collected from Henrietta Lacks in the year around 1951. Okay, these cells, cancer cells, you know, they are immortal. They never age, they never die. Till today, they are cultured in the laboratory. Till today they are dividing. Till today they are dividing. Okay, so the cancer cells, cervical cancer, which she was suffering, an American Negro woman by name Henrietta Lacks. Okay, so these cancerous cells which were collected from this woman, they were abbreviated as HeLa cells. The polymerase activity, these cancerous cells, they divide rapidly. Uncontrolled division will be there. So polymerase activity of the HeLa cells by this DNA polymerase epsilon. Just as an extra information, you try to remember the 5 different types of DNA polymerase involved in mainly eukaryotes. So mainly synthesis of new strand of DNA in 5' different direction, elongation, DNA polymerase delta. Now, what are the other enzymes which are involved mainly in the replication of DNA? Helicase. Helicase is also known as unwindase. Unwindase. DNA is made up of 2 polynucleotide strands which are helically formed, and there are base pairs. Unless and until the DNA is unwinded, uncoiled, on that new strand will not be synthesized. So unwinding nothing but like this. These 2 strands which are helically coiled, that should be unwinded. That should be uncoiled, so that we can get a single stranded tablet strands. So helicase or unwindase, it unwinds the DNA strands. Unwinds or uncoil. Earlier it is coil, uncoiling. Uncoiling means removing the turns of coils which are there. These are the turns, coils. These coils will be removed. As a result, this elongated stretch of what I call single stranded DNA you can get, on that new strand will be produced. So it unwinds or uncoils the DNA strands at what we call ori site. Ori is very important site, origin of replication, the point of DNA from where replication begins, origin of replication. So helicase or unwindase is going to bind at ori site, and it will what we call unwind the DNA strand. How it can unwind the DNA strand? There are the base pairs. Okay, these base pairs are A pair with T, G pair with C, double hydrogen bond, triple hydrogen bond. Okay, so these hydrogen bonds will be melted. Hydrogen bonds will be broken by using ATP. So helicase enzyme make use of energy. Okay, helicase enzyme make use of energy. Right? So it make use of energy that is supplied by the ATP. At a time, complete DNA will not be unwinded, that remember, because it requires more amount of energy. So small stretch of DNA can get unwinded. A small stretch of DNA at a time can get uncoiled or unwinded using the energy. So helicase enzyme can make use of energy. So helicase enzymes can break down, melt, heat the hydrogen bonds present between the base pairs to uncoil the DNA strand. So energy is supplied by the ATP, that remember. Now listen, when The unwinding occurs from, say, this is the ori site, origin of replication. So specific nucleotide sequences will be there at ori site. So in case of E. coli, this site is referred as ori C site. That is origin of replication, ori control site, ori control site. Okay, suppose if this is the ori control site, helicase can come and bind here. Helicase will bind and it start melting the hydrogen bonds present between the base pair, and the DNA strands can get uncoiled. Now, when uncoiling occurs from this side, see here, when uncoiling occurs, when the DNA strand gets separated from here, this side, more number of revolutions are formed. From this end, if you start uncoiling or unwinding, other opposite side, what happens? More number of revolution or turns are formed. This can form supercoils. This can form supercoils. More number of coils are formed. Turns are formed. This can exert tension on the DNA strand. This can exert tension or pressure on the DNA strand. One specific term, 34 angstrom, in DNA. Because of unwinding of the DNA strand from the ori side, there will be more number of turns or supercoils formed opposite end here, this side. Okay, so now the enzyme that is DNA gyrase. Okay, so this DNA gyrase, it relieves the pressure on the DNA strand. It relieves the pressure on the DNA strands caused due to supercoil formation. Supercoil formations of DNA. Okay, DNA gyrase. Gyrase is a type of what we call topoisomerase enzyme. In the case of eukaryote, we say topoisomerase enzyme. In E. coli prokaryote, DNA gyrase. Okay, so it is equivalent to topoisomerase, we can refer. So what DNA gyrase? So here, whenever the supercoils are formed here, DNA gyrase can come and bind to it. It breaks the DNA and reseal the DNA strands. Reseal the DNA strand. It breaks the transient break where more coils are there. So out of the two strands, one strand can be cut. Okay, with the help of this DNA gyrase, it brings transient break where more turns are there. On one of the strands, two strands will be coiled. On one of the strands, it binds to it and it breaks so that that strand can get separated. Okay, it relieves what we call the pressure or the strain tension. Pressure nothing but the tension which is caused. It relieves the tension or pressure on the DNA strand caused due to supercoil. So it brings transient break in the DNA and reseal that DNA strand so that tension can be relieved, relaxed. That is the role of DNA gyrase. In eukaryote, topoisomerase is involved. Topoisomerase 1 and topoisomerase 2 can be involved. So just you remember, it relieves the tension caused due to the supercoil formation during unwinding of the DNA. That is the role of this DNA gyrase. RNA primase. RNA primase is a type of RNA polymerase enzyme. It is a RNA polymerase enzyme. Okay, it catalyzes the synthesis of RNA primer. It catalyzes the synthesis of RNA primer. So RNA primase is nothing but RNA polymerase enzyme which catalyzes the synthesis of RNA primer. We will come to know what is RNA primer. We have DNA ligase enzyme. DNA ligase enzyme. So DNA ligase enzyme, it joins the replicated fragments of DNA. It joins the replicated fragments of DNA, what we call Okazaki fragments. It joins the short replicated fragments of DNA. So small newly formed DNA fragments which are there will be joined and produce what we call lagging strand. When small replicated DNA fragments are joined, it is going to produce lagging strand, that is DNA ligase. So these are some important what we call enzymes which are required in DNA replication. So we have to learn about RNA primer. I told about RNA primer. Without RNA primer, DNA polymerase 3 cannot catalyze the synthesis of new strand of DNA, remember. So RNA primer it is a short segment of RNA. It is a short segment of RNA like this with 5' and 3' and 5' and phosphate group will be there and 3' and hydroxyl group will be there. Okay, so having around what we call 8 to 12 nucleotides in it. This RNA primer is synthesized on DNA template. It is synthesized on DNA template by the activity of this what we call RNA primase enzyme. RNA primase is involved in the synthesis of RNA primer. What is RNA primer? It is a short segment of RNA with 8 to 12 nucleotides having 5' phosphate group and 3' hydroxyl group, OH group. What is the main function of this RNA primer? It plays a very significant role. Without RNA primer, there will not be addition of deoxyribonucleotides by the activity of DNA polymerase 3, so that new strand of DNA can be synthesized. So what is the role of this RNA primer? It provides 3' OH group. It provides 3' OH group for the addition of deoxyribonucleotides by the activity of DNA polymerase 3. By the activity of DNA polymerase 3. Remember this RNA primer, it provides 3' OH. DNA polymerase 3, remember, this DNA polymerase 3 enzyme, it requires pre-existing 3' OH group. It requires already existing 3' OH group so that it can add the nucleotide and synthesize the polynucleotide strand. So that the 3' OH group is provided by the RNA primer. So then you need a specific proteins here in the replication. These proteins are referred as SSPs. They are single-stranded single-stranded binding proteins. They are single-stranded binding proteins. Whenever there is uncoiling of the DNA strands at the ori site, this is the ori site. Helicase has brought the unwinding of the DNA strand. So here, single-stranded structure of the DNA. This is the template strand. This is a template strand. So there is every possibility that these two can rejoin, reseal. When hydrogen bonds are bonded, these two strands can once again get coiled. This is every possibility here. Okay, and to stabilize the structure of DNA, which is single-stranded in this region. In this region, after unwinding, usually DNA is double-stranded, which is stable when it is double-stranded. So here it is single-stranded. Okay, so to stabilize the structure of the DNA and to make sure these two strands remain separate. They remain separate. They do not reseal. They do not rejoin. If they rejoin, it will become like this once again. So to prevent that, here you have specific kind of proteins called a single-stranded binding protein. So single-stranded binding proteins can bind to the single-stranded DNA, which are formed after unwinding at the ori site and stabilize the DNA structure, and they make sure the single-stranded DNA remains separate. They do not allow the rejoining of the separated strands. Lesson. In addition to that, enzymes like phosphorylase enzyme, okay, it is involved in activation of the nucleotides of DNA. Then pyrophosphatase enzyme, It is involved in breaking down triphosphate, nucleoside triphosphate into nucleoside monophosphate. You will come to know when I will discuss this mechanism. So these are also the enzymes which are involved in replication. So this is all about the what we call requirements of the DNA. So the nucleotides are required. The nucleotides are present in the nucleoplasm in what I call eukaryote. Nucleotides are present in the cytoplasm in prokaryotic cell. 4 types of nucleotides, in DNA. That is deoxyadenylic acid, deoxyguanilic acid, deoxycytidinic acid, deoxythymidinic acid, or deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. So these 4 nucleotides are required. DNA template is required. Okay, so these are all various requirements. Remember, we are learning the replication of DNA mainly with respect to what we call E. coli, mechanism mainly involved with respect to Escherichia coli, that is the prokaryotic cell DNA replication. So there, replication occurs in the cytoplasm itself. As the nucleoid, the DNA is presented as naked DNA, without the true nucleus. Whereas in eukaryote, it occurs within the nucleus. Mechanism of semi-conservative replication of DNA. In E. coli, you have double-stranded small circular DNA. So it has a site that is ori C site. Origin of replication. The number of ori is single, remember. In a prokaryotic cell, the number of the ori is single. Single ori will be there. Means replication begins at single site. Origin of replication control site, ori site, to which the enzymes can attach and initiate what I call replication process. So in E. coli, single ori is there, and replication is usually what I call bidirectional. Replication is usually bidirectional. Remember, in case of eukaryotes, the DNA, which is basically linear DNA, at several sites it can have the ori site. It has several ori. It has several ori. Origin of replication. So this ori is must. To this ori, origin of replication, it is must to initiate replication. If this ori is not there, DNA polymerase 3, which catalyzes the addition of nucleotide, will become functionless, useless. Okay, so origin of replication, if this is the DNA strand, from which point the replication should begin, that degree of the specificity is specified by this what I call ori site. There are several ori in case of eukaryotes, that remember, right? So in eukaryotes, John Kerr showed that there are several ori, and replication is bidirectional. There are several ori and replication is bidirectional. It was shown by John Kerr in case of eukaryote. In prokaryote, there will be single ori, okay, from where the replication begins. So this ori is the origin of replication. From there, the unwinding of the DNA strand occurs and it initiates the replication. So this is very important site to initiate the replication. Without this, there is no starting of replication. So that is what, in the case of recombinant DNA technology, okay, in case of recombinant DNA technology, to transfer the desired gene, if this is a gene, okay, a desired gene, this we would like to transfer into, say, a bacterium like E. coli. We want to transfer this desired gene into E. coli, we need a vector. most commonly used vector is plasmid. Okay, so this plasmid has a site, that site is ori site, which can initiate replication. So we need a vector to transfer the desired gene into other organism, into other cell. So the vector used is plasmid, okay, and this plasmid is having self-replicating property independent of the chromosomal DNA. Okay, so because of this ori zone, that's why ori role is very important. That remember. So what are the steps which are involved in DNA replication, especially with respect to E. coli? For our convenience sake, we can have different steps, but usually what we say, the steps of DNA replication: initiation, elongation, and termination. These are the steps which we say. Okay, for better understanding, we can have the steps of replication, initiation of replication, activation of deoxyribonucleotides, unwinding of DNA helix, formation of RNA primer, base pairing, elongation of strand, okay, then termination, proofreading of DNA, So for our convenience sake, we have these steps in DNA replication with respect to a prokaryotic cell like E. coli cell. Okay, so first step, that is initiation of replication. So this is the double standard circular DNA in case of E. coli. So where exactly the DNA replication should begin? So to provide the specificity, where is the site here? That site is called an ori C site. Or just simple, you can call it an ori, origin of replication. Several enzymes can attach to this site and can initiate the replication. Specific nucleotide sequences are there. Will be there, okay, in this part of the DNA. So what we can call ARS, autonomous replicating sequences. Autonomous replicating sequences. Nucleotide sequences are there. Okay, 13 nucleotide long, 9 nucleotide longs. Different reasons will be there. 13 nucleotide long, 9 nucleotide long. Like that, different nucleotide sequences are there, which are the site for the attachment of specific enzymes. Okay, which can initiate the replication process. So here, so ORI site will provide the specificity for the starting of the replication, from where the replication should begin, that is mainly from the origin of replication. So this DNA part has what we call 13 nucleotide sequence long and 9 nucleotide sequence long. Separate boxes will be there in this ORI site. Commonly we call it as autonomous replicating sequences, from where the initiation of replication will be there. Beginning of replication, starting of replication will be there. This is the point, that is ORI, to which later on what happens, helicase enzyme can bind and can bring unwinding of the DNA strand. That will remember. Okay, you will come to know in the subsequent steps. Okay, so this is how the from the ORI site initiation occur. Then the second step, what I said, activation of deoxyribonucleotides. Activation of deoxyribonucleotides. We have four kinds of deoxyribonucleotides. Deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate. These are the nucleotides, monomeric units, building block of polynucleotide chain, that is the DNA. Directly these what I call monophosphate will not be used. They are activated. They are activated. So they are activated, whatever the kind of nucleotide here, they are activated by the addition of phosphoric acid. Okay, using what I call enzyme phosphorylase. Using the enzyme phosphorylase. So what happens here? 2-phosphate group will be added to this. Monophosphate form, 2 phosphate groups will be added. When 2 phosphate groups are added, what they get converted into now? They will be converted into triphosphate form. What will be this? See here, deoxyadenosine triphosphate, deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, deoxythymidine triphosphate. So these will be converted into what we call deoxynucleoside triphosphate forms. Deoxynucleoside triphosphate. These are deoxynucleoside monophosphate. Monophosphate. Okay, deoxynucleoside monophosphate. By the addition of phosphate group given from phosphoric acid, by the catalytic activity of phosphorylase enzyme. Okay, so energy can be used here, and these will be converted into deoxynucleoside triphosphate. So these can act as substrate. Substrate means monomeric unit building blocks. These can act as substrate. They have dual role. Dual role means 2 roles. They have 2 roles. One is they are used as substrate. They are used as nucleotide. Nucleotide in triphosphate form. Say if you take deoxyadenosine triphosphate, this is the nucleoside. These are the 3 phosphate. Nucleotides are added basically in monophosphate form only. But what happens here is there will be breaking of terminal phosphate groups. This result in release of large amount of energy. When these bonds are broken, they release that energy, huge energy. So that energy is used to form hydrogen bonds. To form hydrogen bonds between the base pairs or nucleotide pairs. So the nucleotides which are converted into triphosphate form, they have two roles, two purposes they will serve. One, they are used as substrate. Substrate means the monomeric nucleotide which is joined by phosphodiester bond to form polynucleotide chain. So one, it acts as a substrate, building block, nucleotide, which will be joined. Okay. Another role, as I told, these are existing as triphosphate form. The terminal 2-phosphate high energy bonds, these will be hydrolyzed, these will be broken, and large energy is released. That energy is used to form, establish hydrogen bonds between the complementary base pairs. Complementary base pairs will be there now between the two strands. If A is there, here complementary base pair T. So between them, two hydrogen bonds. Like that. Energy is required for that. That energy comes from the triphosphate nucleosides, which on breakdown release the energy. So two roles, one as substrate and another one the source of energy used for the formation of hydrogen bond. So this is how the four nucleotides which are there, okay, in eukaryote, in the nucleoplasm, in prokaryote, in the cytoplasm itself they are present, and these will be activated. These will be added with the 2-phosphate group, and by the activity of phosphorylase and converted into what we call triphosphate form. So they serve as a nucleotide, monomeric unit, as a substrate, and also source of energy. So these are what I call the two steps which I wanted to discuss today. Tomorrow I will continue, and the real process which we have to learn now from the next step, along with the figure, we have to understand the mechanism of what we call this DNA replication, right? So tomorrow I will continue this important mechanism. Remember, this is very important for 5 marks. This is very important for 5 marks. In theory, explain the mechanism of DNA replication. In NCERT book, they are not given clearly. So try to understand this concept, and in NEET also they can ask what I call many questions on these various concepts, right? So I'll continue tomorrow.